We've seen in previous chapters how challenging it can be to accurately model the processes of translation and transcription and predicting the expressions level in the cell. One additional complexity for modeling translation is the stochastic nature of elongation. In this paper by Peterson and co-workers, they use a stochastic model to describe translation elongation. They model individual ribosomes performing each elemental amino acid extension reaction, and at this granularity they are able to simulate collisions and bottlenecks. Just like cars going down a crowded highway, some sequences are more prone to blockages than others, and protein production is not necessarily linear with respect to translation initiation rates. The phenomenon being captured in their model is an example of biomolecular noise, and more specifically intrinsic noise. In biology, there are two kinds of noise. Extrinsic noise originates from fluctuations in cellular components such as metabolites, ribosomes, and polymerases. These changes in concentration have a global effect on a per-cell basis, but they'll get averaged out over a large population of cells in ensemble measurements. Intrinsic noise results from stochasticity in the biochemical reactions happening at a single gene scale, and these cause two identical copies of a gene in the same cell to express at different levels. Both translation and transcription are known to be sensitive to both intrinsic and extrinsic noise. This paper uses modeling and experiment to describe the issue of traffic on an mRNA and how it is coupled to individual rates of translation elongation. It deals with the issues associated with adjacent slow and fast codons. Random collisions is an example of intrinsic noise because they affect the ultimate expression level of a single gene. Their stochastic model describes translation as distinct initiation, elongation, and termination phases. In each phase, the large footprint of the ribosome bound to the mRNA is taken into account. If a ribosome is already at a certain position of the sequence, then another ribosome is not allowed to exist there as well. Translation starts by binding a 30S ribosomal subunit to codon 1 with a rate Ks, provided that the binding site is not occluded by another ribosome within occluding distance d. The ribosome stays at codon 1 for a time tau to assemble the translating 70S ribosome. After tau, all ribosomes have a probability Rx-dt to move one codon forward, where Rx is the translation rate for the codon at position X chosen from a table of rates for each codon. Movements are accepted only if the distance to the preceding ribosome is larger than d. When the ribosome reaches the end of the mRNA, the peptide is released with a rate Kt. You can play with this simulation online at the shown URL. The interesting variables to play with are the on rate and whether it allows the mRNA to be degraded or not, which is this Start Degradation button. As you start the simulation, ribosomes initiate translation and proceed along the mRNA. You can see how their movements jostle about as they move across due to collisions with other ribosomes. Here's an example of what happens on certain sequences. In these diagrams, each red dot represents a ribosome at some position on the mRNA. At low levels of translation initiation, such as in A, the ribosomes do not accumulate anywhere on the sequence because there is no crowding. However, as the initiation rate increases, the crowding effect gets more exacerbated, and we see a heavy accumulation of the ribosomes around 800 nucleotides into the mRNA. The result of this would be a nonlinearity in the elongation rate, where the expression for beta of translation is no longer linearly related to the translation initiation rate. The biggest claim of the model is an explanation for why there are slow codons at the start of ORFs, which can be seen statistically in natural sequences. The occlusion time will respond to changes in substrate level if the rare codons, for instance GAG, are unsaturated, whereas the fast translated codon GAA is saturated. GAA and GAG are read by the same tRNA, but they are translated with different rates. This allows negative feedback to fine-tune the initiation rate. Slowly translated codons in the beginning of the mRNA will increase the average ribosome distance in the later part of the gene and thereby minimize cues there. So the location of slower and faster codons in an ORF could influence overall translation rate, but also could increase or decrease the distribution of rates observed within a cell. Also, having higher transcription rates combined with a lower translation rate might give less variation. For example, looking back at our Shea-Akers modeling of gene expression, the same steady-state concentration of P can be reached by adjusting the translation rate beta or the transcription rate K. Thus, we could use a stronger promoter and have a higher K while lowering beta for slower translation while still achieving the same amount of protein product.